My name is Scott Trerotola. I'm the Chief of Interventional Radiology at the University of Pennsylvania Medical Center in Philadelphia, Pennsylvania. Post-approval studies can be required by the United States Food and Drug Administration as follow-up after pivotal trials, in this case, the pivotal IDE trial for the Lutonix PTA balloon and drug-coated balloon. And usually they're required because of considerations regarding sometimes safety. In this case, the trial finished at a time when the safety of paclitaxel was being questioned after the Katsanos paper, and the FDA wanted more numbers. They wanted to see just more patients, and that's very common. So it really, although as I'll mention, there really aren't any safety concerns at this point, I think that was probably one of the more compelling reasons for the FDA to want to see more patients in the study. The Lutonix PTA or DCB drug-coated balloon catheter is one of a class of DCBs that are most of which have paclitaxel on them. This one has a dose of 2 micrograms per millimeter squared, so it's at the lower end of the dose range for these devices. It, like all of the other devices, as far as I know, has a low pressure compliant balloon as opposed to the high pressure balloons we use for angioplasty with POBA. It has an excipient, which is polysorbate, which is, again, differs from some of the other devices. So each of these devices is unique in its own way, although all of them have in common that they're using this anti-proliferative drug to try to reduce restenosis. So this post-approval study is a single-arm study looking at more or less the exact same patient population as the pivotal randomized IDE trial did. These are patients with functional AV fistulas that are in use but failing due to any number of factors except for thrombosis, with a stenosis anywhere from the anastomosis to the end of the terminal arch of the cephalic vein, so up until but not including central veins. So the FDA requires interim analyses during the course of the study. This was a planned interim analysis. So the study will enroll 213 patients. This was 81 patients. And because of that interim analysis, we had an opportunity to take a look at interim results, keeping in mind that these are just interim results, but we do have both safety and patency data on those first 81 patients. The findings that we reported for patency were a 6-month target lesion primary patency of 77.6%, which is actually better than we saw in the IDE trial, and is on par with what is being seen in some of the other trials, including the IMPACT trial. So in this more real-world study, with all of the trappings of a serious clinical trial, including a core lab adjudication of lesions and a very careful follow-up of patients, we've seen actually better patency than we saw in the IDE trial, which, as you probably know, was a negative study. So I'm very encouraged that we're seeing results that look like they're benefiting patients. We did see some benefit in the IDE trial in terms of improved time to restenosis, or prolonged time to restenosis, as well as reduced number of interventions to maintain target lesion primary patency. And here we're seeing, some what I look like good patency numbers as well. The safety data are excellent. 100% have met the safety goals. So really, at this point, I don't think there's any concern among any of these devices about safety. So we're seeing good patency, we're seeing good safety, and I think this device does benefit patients. We're at the point when we finish this study, we will have enrolled over 800 patients with Lutonix DCBs in, carefully controlled, designed clinical trials. Some controlled, randomized controlled, some single-arm trials. We have looked in all those trials at lesions all over, and including in the global registry, we had grafts as well. So one thing we have been able to do is sort out whether individual areas of stenosis and or individual stenosis types, as determined by ultrasound, for example, some great work from Kate Steiner in the UK. Also, we are starting to look at the possibility of genetically typing these like we do in cancer, so that we might be able to get a sample and say, okay, this particular lesion is going to respond to a DCB, or this particular lesion needs something else, like a covered stent. This particular lesion may respond just to plain old balloon angioplasty. And that kind of personalized medicine we're going to get to, we're just not there yet.